My name is Herb Schellhorn, and I'm a professor of biology at McMaster University, which is shown in this slide. And today I'd like to talk about some new tools for environmental monitoring that we're developing at McMaster University. So I began to work in this area a few years ago, based on some work that I had done with municipalities to use DNA tools for source tracking purposes to identify, in particular, the way that in the watershed was becoming contaminated from specific sources. And so we worked with government agencies and health officials in doing this. And while doing this work, I realized that there is a larger issue that we can address with an interdisciplinary initiative that includes people that have varying disciplines or varying expertise. And so I formed the McWater Diagnostic Research Initiative to include these individuals, and we submit a proposal to the government for a large grant, which was funded. And this has been a vehicle for our work for the last 10 years. And so the purpose of the McMaster Diagnostic Initiative is to develop low-cost, real-time portable devices so that we can analyze samples in real time for specific biological and chemical contaminants. And so this is an aspirational goal, of course, and we have made significant headway in many of the sub-objectives of this proposal, and I want to talk about a couple of those today. So, of course, as I said earlier, we have to have an interdisciplinary approach because while biologists are curiosity-driven and can help in identifying technology and developing technologies, it's also really helpful to have strong relationships with engineers who have relevant biological expertise. And that's what we've done. We've created a group that has this expertise, and as you can see, there are many engineers, and I've listed engineers as just engineers, but they have specific expertise or work in sub-areas and are world leaders in their particular area. So when I say an integrated approach for water monitoring, I mean we look at the whole process from the point of sample collection through to collection of data, design of specific sensors, integration of material through real-time wireless monitoring to a central server, and then combine this with metadata, including weather and satellite data, to create reports that can be useful to the end user. And so we have members working on each of these aspects. And I want to emphasize that this approach has been, in my view, strongly, the feasibility of this approach has been made possible by several developments, and one is inexpensive DNA sequencing, which you may be familiar with. The cost of sequencing has dropped dramatically over the last 20 years. It cost $20 billion to sequence this first human genome, and today that amount of sequencing would cost about $100. So this has meant that this technology can now be deployed for purposes that were unthinkable just a few years ago, including environmental monitoring. A second is the development of semiconductor-based sensors, which are really useful for detecting specific contaminants, including DNA, for DNA capture devices. And finally, computational advances in wireless transmission and data analysis. So these have made it possible to do things that we just weren't able to do before. We do this in conjunction with partners, both in government and in business. And I've listed a few of our partners on this slide. And this really represents a small group that was, they were partners at the initial stages of our program grant, but we now have expanded this enormously to include many other companies and agencies. So as you can imagine, this kind of integrated approach requires a lot of specialized expertise, and we view this as a platform for training. And I've listed some of the skills that we want our students to develop as participants in this project that will be important for future developments in this field. And so this figure shows the relationship between these skills and our program activities. So for the DNA aspect of this work, we can think of environmental samples as containing really an enormous amount of information that can be used for diagnostic purposes. And when we look at the complexity of DNA in a single water sample, it can be equivalent to several human genomes of information, just an enormous amount of information. And in theory, we can detect each of the possible biological contaminants that can render water unsafe. So we tend to think of E. coli as an indicator of water safety, and that's certainly true. But there are at least 50 waterborne diseases and contaminants that can be present in water, not in all samples, but could potentially be present in water samples. And some of these are difficult to identify because they're just difficult to culture and quantify. But using DNA allows us to determine their presence in an objective way. So this, I said, is partially driven by reduction in DNA sequencing costs. And I just want to show you this graph that really demonstrates how dramatically DNA sequencing has dropped. And this is around 2007, 2008, and this is on a logarithmic scale. So you can see that sequencing that cost $100 million in 2001, just 20 years ago, now costs about $1,000. It's even less, actually. But this is largely because of a change in the sequencing technology. So it's dropping much faster than the computer, which is governed by Moore's law that you may have heard of. And you can look that up if you want more information. But it's really dropped a lot. So we can think of DNA in environmental samples as strings of information, and that's what I've depicted here, a large gray ball. And traditionally, we use, for source tracking purposes, 16S RNA, which is a universally distributed gene that's found in all organisms, but the sequence is a little different. And because it's a little different, we can use this for diagnostic purposes. And I've represented this as a series of gray boxes because the sequences are just a little bit different. And we have to use complex analysis and identify these against large data sets. And depending on the algorithm we use or the methods we use for identification, we can get variable results. One user might get a slightly different result than other users. So these are non-unique identification solutions. However, at McMaster, we've developed and this is really the work of Rad Gupta, who is in our biochemistry and is part of our McWater team. He's identified conserved signatures for thousands of organisms, and these sequences stand out in DNA. They're present not universally, but they are found within a specific group that may be of interest. So we can have one for Campylobacter, we can have one for cyanobacteria, we can have one for E. coli, and in many cases we have more than one. And so these conserved signatures are easy to identify because I've shown them as brightly colored, but they're easy to distinguish from other sequences. And so this leads to simple identification using very few DNA sequences and a single identification. And so this is important from a public health point of view because now you have a technology that gives a single result. And it's really independent of the end user. It doesn't matter whether the end user is highly trained or poorly trained, they're going to get the same result. And that's helpful because we want unambiguous, robust results for public health decision making. So I'll just mention that some of our previous and current projects that are ongoing, and I won't go, these all have specific aims, but we really started in the region of Niagara, and you can see Niagara Falls here. Here is Hamilton, where we're located, and Toronto is up here. So this is about 80 kilometers from Hamilton to Niagara Falls. So we had a contract with the region of Niagara to do specific types of projects there for source tracking, and this was really important initial work. Then we worked with the Conservation Authority and looked at this 80,000 square kilometer watershed We paired with their sampling and did DNA analysis. And then there was a small issue in the town of Wainfleet that had a long time boil water advisory, and we worked there as well. And we conducted a phosphate reduction study in Essex County near Detroit, actually. You can see Detroit here, and this is an important area of Ontario for agriculture. Currently, we're working with the city of Toronto, which is Canada's largest city, has several recreational beaches along the Lake Ontario shoreline, and we're conducting the feasibility of using an enterococcus test for determining beach safety, because this will take 4 hours rather than overnight or 2 days, as is the case with traditional E. coli counts. And so this will allow the city to make same-day beach opening and closing decisions, which is really helpful, because people want to know on the day they go to the beach if it's safe or not whether it was safe yesterday. So we're working on that. That's a 2 or 3 year project. Well, it's become 3 years because of COVID. And we're doing other related projects. And I'm just talking about our group. The McWater group is engaged in many different types of projects with many partners. So this just shows some of the papers that we've published on this project. There are others now that we've published, and we've obtained new project funding that'll sustain our work for a few years. And I just want to acknowledge that this work could not be done without a lot of partners. I haven't really mentioned too much about the external partners, but these are the key people. Rad Group I mentioned, Tom Edge at Environment Canada, and the my lab and the Gupta lab, and of course getting funding from agencies is very important. So I'd like to thank the organizers of the Canadian Network Spotlights for giving me the opportunity to tell you a little bit about our work at McMaster. And I think this kind of work is applicable in Austria and other jurisdictions as well. And so I think it's very important. Thank you. I just want to leave you with our website if you have more information at mcwater.org. And this describes our group, and the significant information that's contained in this site, I think, is in our meetings section, which describes some of the meetings we've had and the talks. And so you may find that helpful or interesting if you have more interest in this initiative. Thank you very much.